Hello everyone, welcome. My name is Peter Engel, and today I will be proposing my idea for creating a patient registry and implementing biosimilar gene therapies to improve equitability in the treatment of sickle cell disease. My guiding research question is, how can equity be improved in providing access to new revolutionary sickle cell disease treatments for the disproportionate number of sub-Saharan African patients? My two main methods of achieving this goal are setting up a patient registry in sub-Saharan Africa and giving access to the newly developing gene editing treatments for sickle cell disease through the use of biosimilar gene therapy. So, what is sickle cell disease? Well, sickle cell disease is a hereditary genetic disease that is caused by a single base mutation. For some clarification, hemoglobin is an essential oxygen-carrying protein that allows red blood cells to deliver oxygen to the body. And in sickle cell mutation, as the name suggests, the incorrectly produced hemoglobin causes the red blood cell to take on a sickled shape, as you can see in the image on the right. The cells also become firm, adhesive, and unable to carry oxygen properly. This can cause very severe anemia, pain crises, tissue death, and even stroke. The equity concerns led me to focus my plan on sub-Saharan Africa. In 2015, more than 1.5 million babies were born with sickle cell anemia in Africa. Around 80% of all sickle cell disease cases are located in sub-Saharan Africa. Up to 3% of the population may be affected by sickle cell disease. The disease is associated with a childhood mortality rate of over 50% for children born with sickle cell disease. That clearly shows that resources to properly treat the disease are unfortunately not available in these regions, and thus children are dying at alarming rates. Before I get into depth about my proposal, I want to briefly describe new revolutionary gene therapies for sickle cell disease. Two companies, Vertex Pharma and Bluebird Bio, are developing CRISPR gene editing therapies to cure patients of sickle cell disease by correcting the faulty beta-globin gene. The editing happens ex vivo, or outside the patient's body, in their own stem cells, known as HSPCs. After successfully editing the gene, the cells are then reinfused into the patient. The diagram shows the step-by-step process for this. So HSPCs are collected, grown, edited, and eventually, after the patient goes on chemotherapy to kill off the last of the defective cells, the edited cells are reinfused to hopefully cure the patient. The first aspect of my proposal aims to create a patient registry in sub-Saharan Africa. The registry will collect genetic, phenotypic, and case-specific data from the large population of sickle cell patients. This will be formatted very similar to the Surveillance Epidemiology and End Results Program, also known as SEER. SEER has been very successful in facilitating advancements in cancer research and treatment with their well-structured and useful bioinformatics data, and my goal would be to do something similar with my registry. The second aspect of my proposal aims to improve access to the revolutionary gene therapies I mentioned earlier through the use of biosimilar gene therapies. Biosimilars are a near copy of the original treatment, but significantly cheaper as it's not through the original company. So the goal is to get a license to the patents from Vertex Pharma and Bluebird Bio for sub-Saharan Africa so that you can manufacture for a much cheaper price. Right now, the therapies in development are estimated to cost around $2 to $3 million, and my goal is to expand access by cutting costs. My inspiration was that during the AIDS epidemic in the early 2000s, the generic manufacturer Sickle made copies of new revolutionary antiretroviral cocktails and sold the life-saving drugs for $300 instead of $12,000 per year to African charities and governments. However, it's important to consider that gene therapy is a much more complex therapeutic procedure which requires hospitals, labs, and drug delivery technology and more. This project aims to create a roadmap for overcoming the issues that need to be solved such that patients in need can be cured. I'm looking forward to your questions.